as I was discussing, that in all the complement pathways, as I had mentioned in the last lecture, that there is a cleavage of the complement proteins, right? And these cleavages are initiated by several proteases, and serine proteases are also one of them, right? So,
Ma'am?
am I audible? Hello. So the answer to this question, question number 2, assignment 9, it is all the
Yeah.
complement pathways, because I told you, like, in all the complement pathways there is a cleavage of the complement proteins, right? For example, 3A, complement 3 protein cleaves into complement 3 and 3B, 4 cleaves into 4A and 4B, and 5 cleaves into 5A 5B, like this. So all the cleavages, they take place by the help of some proteases, right? And serine proteases are also one of them. So not only the serine protease, the membrane associated serine proteases, which are involved in the which are involved in the lectin pathway, but also the in all the other complement pathways, these complement proteins are cleaved by some or other complement, some or other proteases which cleave these complement proteins. So because of which this is the answer, is all the complement pathways. Yes, Vandana. Vandana, did you want to say something?
Ma'am just wanted Mr. Rajkumarji to switch the mic. That's it.
Oh, okay. Yes. So I think I made this clear to you, that in all the complement activation pathways there is a cleavage. The essential component of the complement activation pathway is the cleavage of this complement protease, right? And the cleavage is mediated by proteases. One of them is serine proteases. So serine proteases are important for all the complement activation pathways, classical, alternative, as well as lectin binding pathway, right? Okay, so let's start today's session, week 10 live session of immunology. Good evening. Yes, Rajkumar.
7th question, ma'am.
Seventh question. Okay,
7th one.
I'm checking.
Okay, ma'am.
Negative regulation of the complement pathway is essential for the protection of self tissue. Which among these contribute to the negative regulation? Factor I, factor BH, C4BP, all of the above. So yeah, please tell me, what is your question? The answer should be all of the above, because if you remember that after this C3 convertase is formed, right? So there are several ways, like there are several pathways by which these complement regulation, like the regulator proteins can stop the complement activation, right? And for that, factor I is one factor which actually takes action after the C3 convertase, the C3BBB or C4B2A, after they have formed, or before they had formed, right? So what it does is that it cleaves cleaves the proteins C3B and C4B, so that the proteins C3B and C4B, they cannot be activated anymore. So the two C3 convertases, the C3BBB or the C4B2A, these two convertases are inhibited to form by the factor I. Okay, so factor I cleaves this C3B and C4B to inhibit the formation of C3 convertases. Next is factor H. factor H, what it does is that in the alternative pathway, so what happens that the C3b um, C3b is associated with factor B to cleave factor B into BB, and then this C3b forms this C3 convertase, right? So the role of factor H is to it prevents the association of factor B with C3b, all right, so that this C3 convertase cannot form. Next is this C4 binding protein. What it does is that it interferes the assembly of the C4b with C2a. Okay, so C4b binding protein does not allow the C4b to associate with your C2a, so that this C4b2a complex cannot form, which is a C3 convertase again, right? So these are the several steps where this negative regulation of complement activation can take place. So the answer is all of the above.
Okay, thank you, ma'am.
Okay, all right. So let's begin our session today. Welcome you all to the live session of week 10, and in today's session we will be discussing about hypersensitivity. Okay, so what is hypersensitivity? So we know that whenever there is a pathogen invasion in our body, there is a immune reaction that generates. This immune reaction can generate from the it begins with the innate arm of the immune system, and then later on the humoral immune response also develops. But what is this hypersensitivity? So from the name you understand this hypersensitivity is nothing but a heightened response to any pathogen, to any foreign antigen. Okay, and this heightened response obviously means that it's a hyper response, or it is a response which is not so desired. Okay, it is not a very wanted kind of response from the immune system, but the immune system becomes too sensitive and it reacts in a like in a furious way, right? That is to like simply explain hypersensitivity. Okay, so hypersensitivity can generate from uh, several types of environmental pathogens, several types of innocuous antigens, or some other antigens which may come in our body, and then the immune system generates this hyper response. All right, so there are several types of hypersensitivity reactions. So let's see. Okay, so there are mainly 4 types of hypersensitivity reactions: type 1, type 2, type 3, and type 4. All right, so what is type 1, and like what are the different types? Why are they differently mentioned in different types? So first thing is, hypersensitivity reactions can be either mediated by the humoral branch of the immunity, or it can also be mediated by the cell mediated branch of the immunity. Okay, so the type 1, type 2, and type 3, these 3 types of hypersensitivity reactions are mediated by the antigens and antibodies. So type 1, type 2, these 2 types of hypersensitivity reactions are mediated by the antibodies, whereas type 3 hypersensitivity is mediated by antigen antibody complex. All right, so these 3 types are mediated by the humoral branch of the immunity, the antibodies, or the antigen antibody complex, whereas the type 4 type of hypersensitivity is mediated by the T cells. Okay, so it is a cell mediated hypersensitivity. So let's have a look at what are the different types of hypersensitivity, why are they, what is the difference between them. So let's look at type 1 hypersensitivity in the beginning. So type 1 hypersensitivity is a very common type of hypersensitivity, and whenever you have these, for example, pollen allergy, dust allergy, some people also have like cold allergy, right? Most of these allergies, they are nothing but hypersensitivity reactions. Okay, so which is why the antigens which generate this kind of hypersensitivity reactions are also known as allergens. Okay, so type 1 hypersensitivity is mediated by IgE. Okay, the antibody IgE mediates the type 1 hypersensitivity reaction. Now let's have a look at how this happens. So the dendritic cells, the dendritic cells, they whenever there is a suppose there is an antigen that has uh, come into our body, suppose this is a pollen, or suppose this is a dust particle, right? So what happens, that dendritic cell, they present this antigen on their surface, right? They present this antigen on their surface, and they can activate CD4 plus T cells, right? So they can activate this CD4 plus T cells by the antigen that it presents, right? And once this CD4 plus T cell is activated, it can be differentiated into a T helper 2 type of cell, right? Th2 type of cell. Now why Th2? Because Th2 can produce one cytokine which is IL4, right? IL4 cytokine, which helps in class switching of the B cells, right? So now the B cells will be able to produce different classes of antibodies, right? So IL4 mediates this class switching of B cells, and due to after this class switching, B cells now can either produce a plasma cell. The plasma cell is what? The plasma cell is the cell which will produce different types of antibodies now, and the or it can also generate the memory B cell, right? So from an activated B cell, a memory B cell will generate, or a plasma cell will generate. Now the plasma cell, they will now produce this antibody IgE. Okay, now what is the role of this IgE? Now the IgE actually comes and bind to the surface receptors, to several cell surface receptors on the mast cells or the basophils, right? So this type of hypersensitivity, the type 1 hypersensitivity, is primarily mediated by these granulocytes like mast cells or basophils. So these are the granulocytes. Why? Because what is the role of these granulocytes here? So once this IgE comes and bind to the Fc receptors, the Fc receptors on the surface of the mast cells or the basophils, then what happens, these, like, so the Fc receptors are what? These are the receptors which can recognize the Fc region of the IgE or the other types of antibodies, but there are specific Fc receptors for specific types of antibodies, right? Now once these IgE molecules, they come and bind to the Fc receptors, there's a downstream signaling that takes place, right? Because of which there's an event that occurs, is that degranulation of the mast cells. Okay, so mast cells are degranulated. So the granules of mast cells, they contain several inflammatory molecules, as previously mentioned also, like the histamines, prostaglandins, leukotrienes. So these are the granules which now come out of the mast cells, and these molecules, these granules, can now cause vasodilation. Vasodilation is something that you know, it is the increased permeability of the vasculature, right? And also it can help in contraction of the smooth muscle. So in these ways they help in enhancing the inflammation, right? So this is what goes on in the type 1 hypersensitivity. Now if we have a closer look at what happens when this IgE comes and binds to the Fc receptors of the mast cells. So let's have a look here. So this is the phenomena that occurs when uh, um, when the IgE molecules comes and binds on the mast cells and the Fc receptor. Now I told you that there is a specific Fc receptor for each class of antibody. For the IgE, there is this Fc epsilon R1. So I'm writing it here. Fc epsilon R1, which is the Fc receptor for IgE. Okay, so IgE comes and binds to this Fc receptor. All right, and then what happens? This IgE is bound to the allergen already, right? So this particular thing is the allergen or the antigen, right? So once the IgE molecules they bind to the antigen, they now come and bind to the Fc receptor on the surface of the mast cell. And these red ones are the Fc receptors, right? Now after they bind to the Fc receptor, so these are the high affinity Fc receptors that are present on the surface of the mast cell. And once the IgE molecules bind to the Fc receptor, it causes cross-linking of the Fc receptors. So this term I was mentioning in my previous lecture also, cross-linking of the receptors. So the receptors come close together. And once this cross-linking happens, it generates, so first one, what happens? IgE binding to Fc receptor, followed by cross-linking of the receptors. And once this cross-linking occurs, there is this activation of protein tyrosine kinases which are associated with the Fc receptors. So this is the PTK or the protein tyrosine kinase. Okay, so protein tyrosine kinase is activated, and this protein tyrosine kinase, it phosphorylates something. What does it phosphorylate? It phosphorylates phospholipase C. So this is the phospholipase C. So this protein tyrosine kinase, it comes and phosphorylates the phospholipase C. Phosphorylates the phospholipase C, and once this phospholipase C is phosphorylated, so what it does is that it again phosphorylates PIP2. So what is PIP2? PIP2 is inositol bisphosphate. Okay, phosphoinositol bisphosphate is PIP2. This PIP2 is phosphorylated by the help of this phospholipase C, right? And it phosphorylates PIP2 to form IP3 or inositol triphosphate. Inositol triphosphate, and another molecule that forms, that is diacyl glycerol or DAG. Okay, so here you can see that PIP2 is phosphorylated to form IP3 and DAG. Now these IP3 and DAG, both molecules have several roles in the upcoming process. So what is the role of IP3? So what IP3 does is that IP3 comes in effects on the endoplasmic reticulum. So what does it do? In the endoplasmic reticulum, there are these calcium molecules are released because of the effect of IP3. The calcium molecules are released from the endoplasmic reticulum. All right, and what is the role of DAG? DAG activates one kinase, which is protein kinase C. Okay, so what DAG does is that it activates PKC, and what IP3 does is it releases calcium ions. Okay, now these calcium ions along with the active PKC, they help in the contraction of the microtubules of the cell. Okay, so when the microtubules are contracted, it will help the granules of the cells to move towards the membrane. Okay, and these granules contain the inflammatory molecules like histamines, the prostaglandins, all these amines, right, vasoactive amines. These are contained in the granules, and these granules now start to move towards the membrane. Now one more thing that happens here is that due to this activation of the protein tyrosine kinase, one other event also occurs, that is the phosphatidylserine, one lipid molecule of the membrane, is now converted to phosphatidylcholine. Okay, so phosphatidylserine is converted to phosphatidylcholine. And what is the role of this kind of conversion of the lipid molecules of the cell? You know that lipid molecules determine the fluidity of the cell, right? So the phosphatidylcholine, once it is converted to phosphatidylserine, once it is converted to phosphatidylcholine, it allows the membrane to be more fluid, and it allows the opening of the calcium channels, the membrane bound calcium channels, because of which what happens, more and more calcium ions come into the cell. Okay, so there is this requirement of calcium ions inside the cell, and because of this inflow of the calcium ions across the membrane, more microtubule contraction can take place, and which is why the granules now start to move towards the membrane, right? And by exocytosis, so the granules fuse with the membrane, and by exocytosis these contents of the granules are now released outside. What are these contents? These are mainly histamines and other vasoactive amines, right? So this is the whole process that occurs, which ends with granule exocytosis, leading to release of histamine. Now this histamine, now this whole, first of all, let me tell you that this whole process that is occurring here, this whole process is known as degranulation of the mast cells. Okay, so this degranulation now causes release of these histamines, and because of this release of histamines, now these like these vasoactive amines, now they start to bring in more and more due to vasodilation, so they bring in more and more immune cells to the site of action, and it causes a local inflammation, right? And this inflammation can be like itching, it can be like redness, it can be swelling, right? So these kind of events occur in this hypersensitivity reaction, and you can also have sneezing, you can also have cough and cold, right? This kind of allergic responses we face because of these histamines, right? And of course from this so this is all about this type 1 hypersensitivity reaction, and from this you must understand that the causing molecule of this type 1 hypersensitivity is the histamines, right? So the principal molecules are the histamines, and if histamines can be blocked by if histamines cannot interact with the receptors, if the binding of the histamine to the receptors can be blocked, then of course this type 1 hypersensitivity can be reduced. So that is what is done in type 1 hypersensitivity. The antihistaminic drugs are administered so that this kind of hypersensitivity reactions can be curtailed. And what it does is that it does not allow histamine to bind to its receptor and cause further inflammatory responses, right? So this is all about the type 1 hypersensitivity reaction. Anyone has any question? All right, if you don't have any question, let's move on to type 2 hypersensitivity. Now the type 2 hypersensitivity, it is mediated by one particular process which are called the antibody dependent cytotoxicity, right? So it is ADCC. Okay, so type 2
antibody dependent cellular cytotoxicity or cell mediated cytotoxicity.
sorry,
ABCC was said as antibody dependent cell mediated cytotoxicity.
exactly antibody dependent cell mediated cytotoxicity, exactly. So this is type 2, this is the type 2 hypersensitivity reaction. And type 2 hypersensitivity reaction is like a very classic example of type 2 hypersensitivity reaction is erythroblastosis fetalis. So what is that, we will discuss. But type 2 hypersensitivity reaction, remember that it is mediated by IgG. Okay, so type 1 hypersensitivity was mediated by IgE, whereas type 2 hypersensitivity is mediated by IgG. And what IgG does is that what is the function of the antibodies? The antibodies can help in opsonization, the antibodies can help in complement activation, right? So this is what it happens in this kind of hypersensitivity reaction also, that the antibody generates these two types of responses. Either it can be complement activation or it can be antibody dependent cell mediated cytotoxicity. So what happens here is that the antibodies, the IgGs, they bind to the receptors, right? And then it initiates the killing of the target cells. Okay, so this antibody dependent cell mediated cytotoxicity can occur when like there is, suppose a particular antigen that has come into our body, and then immune system generates a heightened response against that antigen. All right, so type 2 hypersensitivity, as I was mentioning, that it is a very classic example of type 2 hypersensitivity is erythroblastosis fetalis. So let's discuss what it is. Okay, so erythroblastosis fetalis is the condition when a Rh negative mother gets pregnant for the second time with a Rh positive fetus. What it means, first of all, you know that our blood groups, it has Rh antigens, right, which determine whether our blood group is positive or whether our blood group is negative, right? So these Rh antigens are present in the positive blood groups. So for example, A positive, O positive, AB positive, or B positive. But this Rh antigen is absent in the blood group of these Rh negative people, right? Now this Rh antigen, when suppose a mother when she conceives, and she, remember that the mother is Rh negative. Okay, now when she conceives for the first time with a Rh positive fetus, so the fetus, the baby, is Rh positive. Now what happens, this Rh antigen might come into the mother's blood and create and generate antibodies, but this does not usually happen during the pregnancy, because there is very little mixing of the fetal blood with the mother's blood. Okay, so almost there is no mixing between the fetal blood and the mother's blood, which is why the Rh positive fetus, it remains safely in the womb of the Rh negative mother. However, during the time of delivery, a little mixing of the blood occurs. Okay, which is why this Rh antigens from the fetal blood might come into the body of the or the blood of the Rh negative mother. Okay, so when this happens, so naturally the Rh positive antigens, they come in the blood of an Rh negative person, and naturally this is considered as an foreign antigen, and the mother's body produces antibodies to tackle with that particular antigen, right? Now, not only it will produce antibodies, but it will also, after this activation of the B cells, it will also produce memory B cells, right? So the plasma B cells will produce the antibodies, whereas the memory B cells will remain in the body of the mother. Now this time, the first time of this like the first pregnancy, during the first pregnancy, there is usually no such problem, and and the body of the mother generates an immune response to the Rh antigen, but it does not cause any other problem. But the problem occurs when the mother is pregnant for the second time with a Rh positive fetus again. So now what happens, the mother's body has all these anti-Rh antibodies, right? So the antibodies which can recognize the Rh antigen. Now what happens, now these antibodies, if they pass this barrier of the placenta, and if they are passed on to the fetus, yes, I am still on the first slide. Yes. Okay, so if these anti-Rh antibodies, if they are passed on to the fetus, so what it will happen is that these anti-Rh antibodies will recognize the Rh antigen, right? And this will cause a hemolytic condition. So you understand that, like, all in the blood of the fetus there will be lots of Rh antigens, and these Rh antibodies will try to kill all these Rh antigens, and this will cause a severe condition in the fetus, okay, which might also lead to death of the fetus. This is actually known as erythroblastosis fetalis, this particular condition, that when during the second pregnancy, the antigens, not the antigens, the antibodies, the anti-Rh antibodies from the mother, from the Rh negative mother, passes on to the fetus, and it generates a hemolytic condition in the fetus. Okay, this is known as erythroblastosis fetalis. So now you will obviously ask, like, how to overcome it, because a Rh negative mother can give birth to a Rh positive baby, and will it cause problem every time? So thankfully we have a cure or a treatment to this particular condition, and that is mediated by some antibodies which are called ROGAM. So ROGAM is what? ROGAM are antibodies against the Rh antigen, right? So nowadays what is done is that the doctors do not wait till the second pregnancy to arrive. Whenever a mother is actually pregnant with a Rh, whenever a Rh negative mother is pregnant with a Rh positive fetus, so during the time of delivery, just after the delivery, within 2-3 days, the mother is injected with this antibody called ROGAM. Why is it done? Because these antibodies are anti-Rh antibodies, right? So they will generate or they will neutralize, or they will opsonize the Rh antigens. And now if these antibodies are injected into the body of the mother, then what will happen? The memory B cells of the mother will not be produced. All right, so these memory B cells, if they are not produced, they will not retain the memory of an Rh antigen invasion, which is why when the mother will be pregnant for the second time with the Rh positive fetus, there will be no memory B cells to generate anti-Rh antibodies and to pass it on to the fetus. Alright, so because of which this mother has to be treated or has to be injected with these ROGAM antibodies as soon as possible after the delivery, so that the B cells cannot be generated. If these antibodies quickly go and kill the Rh antigens and then destroy the Rh antigens, then what will happen? The immune system will think that yes, this infection is clear, so therefore it will not activate the B cells any further and it will not generate any memory B cell. Alright, so this is a very classic example of type 2 hypersensitivity, which are mediated by the IgGs, right, and often seen in case of Rh negative mother being pregnant with Rh positive fetus. But thanks to the medical technologies, now we have a antibody which can help to like counteract this particular situation. Okay, so am I clear till this?
Yes, ma'am.
Okay, all right, so now let's move on to the other hypersensitivity reactions, for example the type 3 and type 4. Now type 3 hypersensitivity, as I mentioned already, the type 3 hypersensitivity is mediated by antigen antibody complex or the immune complexes. Okay, this is type 3. Now once this so type 3 hypersensitivity, once there is a formation of an antigen antibody complex, right, it will generate many different kinds of responses. One of them is the activation of the complement pathway, right? So the complement pathways are activated. Now because of this the generation of these immune complexes, the antigen antibody complexes, one thing that will occur is that the complement pathway will be activated, and because of this activation of the complement pathway, there will be several complement proteins which will be cleaved, right? So we talked about this cleavage of C3 to C3b, C3a, then C4 is cleaved into C4a and C4b, the C5 is cleaved into C5a and C5b, right, so on. And these A's, these 3a, 4a, 5a, these are all the soluble components, and the these are the soluble components of the complement pathway. And what happens here is that this complement activation takes place in several ways. One of them will be the membrane attack complex formation, right? So the C3b, it goes and binds to several receptors, the complement receptors present on the neutrophils, right? Also these soluble cleavage products, the C3a, C5a, C4a, these all come and bind to the receptors of the mast cells or the basophils, right? And as I mentioned previously also, that these mast cells or the basophils, they are granulocytes. Okay, so they contain the granules for which contains these vasoactive amines like histamines, right? So once these C3a, 4a, 5a, these complement cleavage products, they come and bind to the receptors on the mast cells of basophil, what happens is that it again causes this degranulation of the mast cells. And not only that, these C3A, 4A, 5A, these molecules can also act as chemoattractants. This I already mentioned in my complement classes also, where these proteins, these soluble forms of these complement proteins, they also help in the chemoattraction. So they attract more and more neutrophils to the site of action. Okay, so they cause this vasodilation, right, so that the vascular permeability increases and these neutrophils can come to the site of action. All right, so here you can see that this complement pathway is acting in several ways. One is the binding of C3b, okay, to the complement receptors on the neutrophils. One is by the chemoattraction of the more and more neutrophils to the site of action. Other is mediated by the granulocytes, which is by the release of histamines, right? So there are several ways by this complement pathway activates, and what it does is that it finally releases many lytic enzymes, right? And these lytic enzymes, so what do? They cause these um, phagocytosis of the antigen antibody complexes, right? This is a this response, this type 3, this is actually the basis of the type 3 hypersensitivity response. And one common example of type 3 hypersensitivity is serum sickness. So what happens in serum sickness? Suppose we have a snake bite. Okay, so when we have a snake bite, we are usually administered with antivenom, right? What is this antivenom? This antivenom basically contains antibodies against the antigen which is present in the snake venom. Okay, now how is this antivenom prepared? The antivenom is prepared by injecting the snake venom in several animals, right? For example, horse is a very common animal in which this antivenom is generated. So for once there is a snake bite, we cannot wait for the antibodies to be produced by our own immune system, so we need special help. So we provide this antivenom which contains the antibodies which will quickly neutralize the antigen of the snake venom, and it will help save the person. So what happens is that this antivenom is produced in horse, right? So when this antivenom is extracted from the horse, along with the horse serum, there will also come some antigens from the horse itself, right? These antigens can then generate an immune response in the body of the person where this who is being injected with this snake venom, who is being injected with this antivenom. So what is happening here? Suppose this person is injected with antivenom, and this antivenom also contains some horse antigens. Now in response to this horse antigen, of course this person will generate an immune response. So this immune response is known as serum sickness, and it can generate a local as well as a global kind of response. And this response is initiated by these formation of antigen antibody complexes, right? And this response is not a required response, as I mentioned about hypersensitivity overall, that these horse antigens, they will not cause any harm to the body, but since it is a foreign antigen, since it is foreign to our body, our immune system generates a response to the horse antigen, and it results in serum sickness. Okay, so along with the treatment of the snake bite. So this is all about type 3 hypersensitivity. Any queries from this? Okay, if not, we will move on to the last type. I think it's antibody that for neutral. Shashwato, I did not actually understand your query.
And the snake venom contains antigen, and the antivira that was collected from horse contains antibody against that antigen.
Yes.
And when that is injected into our body, that will contain antibody to that antigen.
Yes, exactly. So the snake venom is having the antigen of the snake. To counteract these antigens from the snake, we inject with antivenom. This antivenom is taken from a horse. So this antivenom is generated in a horse by injecting the venom in the body of the horse. And when this horse produces these antibodies against the snake venom, so these antibodies are collected from the horse, and when this horse serum is injected in our body, these antibodies against the snake venom will now neutralize the antigens which are now has infiltrated in our body due to snake bite. Now along with this antivenom, along with these antibodies, there will come some horse antigens which will uh, cause problem, which will cause this type 3 type of hypersensitivity in our body. Okay, and to tackle that, so the horse antigens have to be removed from the antivenom so that this kind of hypersensitivity reactions cannot generate. Is that clear?
Yes, ma'am, it is clear. So it is not antibody. It is serum sickness is because of the antibodies in our blood because of the antigens present in horse serum.
Exactly.
Thank you so much, ma'am. Thank you.
No, it's not the antibodies. Oh, sorry. This is what you wanted to ask actually. No, it's not the antibodies. Rather, it's the antigens that are coming from the other animal, because like whatever the antibodies the so basically what is happening, the horse is also producing some antibodies in its serum, right? So in a healthy horse also, this particular horse will produce some antibodies always. It will have its serum will always have some antibodies, right? But these antibodies, when it will come in our body, it will be treated as a foreign particle to our body, right? So they will be treated as antigens. Okay, so therefore it will generate this immune response. So in a way this antibody is also correct, because it's like the antibodies are also treated as foreign antigens, the horse antibodies, not our antibodies. Okay, tell me if that is okay, that clarifies everything.
Yes, ma'am. Thank you so much, ma'am.
Okay. All right, so now let's move on to the last part, which is the type 4 hypersensitivity reaction. Now the type 4 hypersensitivity reaction is called the delayed type hypersensitivity. Okay, and initially it was thought that first of all it is mediated by T cells. Okay, and initially it was thought that it is mediated by TDTH type of cell, which is like T delayed type hypersensitivity cells. But later on it was found out that this is nothing but these delayed type hypersensitivity T cells are nothing but a subset of T helper 1 cells. Okay, and this is initiated by the Langerhans cells, or the neutrophils, or the dendritic cells that are present in the epidermis. So the Langerhans cells, they present this antigen, right? They present this antigen to the T helper 1 cells, and the T helper 1 cells recognize this particular antigen, and it gets activated, right? And when the T helper 1 cells gets activated, they of course produce many they of course produce many cytokines. Okay, so the activation of the T helper 1 cells produce many cytokines like TNF alpha, interferons, IL-3 or granulocyte macrophage colony stimulating factor. Like these cytokines are produced, and these cytokines, what it does is that they activates the macrophage. Okay, the tissue macrophages, they get activated by this kind of release of the cytokines, and this macrophage now mediates the type 4 type of hypersensitivity. This generates a delayed type of hypersensitivity. Okay, and so what actually happens, these macrophages, when they get activated, they also produce some other cytokines, and it creates a local inflammation, like some redness, swelling, those things you can observe in a local site where these hypersensitivity reactions take place. Okay, now one classic example of type 4 hypersensitivity is the Mantoux test. Does anyone know what is this Mantoux test?
Yes, ma'am, it is a traditional test that was done to diagnose maybe tuberculosis.
Tuberculosis, exactly. So it is a very old test which was used to diagnose tuberculosis, whether a person has been previously exposed to Mycobacterium tuberculosis or not, right? So what is done in case of this Mantoux test is that the tuberculin protein, or the protein peptide derivative, the tuberculin, is injected uh, just under the skin of a patient, right, of somebody whom you want to diagnose whether this person has been exposed to TB or not, or whether the person has TB or not. So the tuberculin is injected under the skin, and what happens that you have to wait for 2 to 3 days, and you have to check whether there has been a swelling at this particular site where this tuberculin was injected. Okay, so the tuberculin is suppose it is done in the on the arm, and it is injected, and after a few days, if you report a swelling, then it will be confirmed that yes, you had been you had been infected with tuberculosis in the past, or you are now infected with tuberculosis, right? So this is Mantoux test. And why do we have to wait for 2 to 3 days? Because as I told, that type 4 hypersensitivity is a delayed type hypersensitivity, so it is not, it does not start immediately, rather it takes some time to activate the macrophages, which is why it is a delayed type hypersensitivity, right? So the Langerhans cells will first present this antigen to Th1, and then finally this Th1 cells will activate the macrophages, and the macrophage will produce these inflammatory molecules, or it will create this local inflammation, swelling or redness. So to that to happen, it will take some days, and this is why it is a delayed type hypersensitivity, because it is mediated by the cell mediated branch of this immune system, which are the T cells, the T helper 1 cells, right? So Mantoux test is an example of type 4 hypersensitivity, which can be used to detect whether a person has been exposed to TB or not. Yes, Vandana?
Ma'am, I was unable to get what you call those cells as Langerhans,
Langerhans,
like
yes.
islets of Langerhans.
Yes, yes, exactly.
Okay, and ma'am, these days Montu is not done to detect TB. It is an obsolete test.
Exactly.
Rather, on the contrary, some people in dermatology they use this to check whether the immune system is functioning properly or not.
Exactly, because the Mantoux test is not a very confirmatory test, because whether you had been infected with TB in the past or whether you're currently infected with TB, it's not a very determining test. So which is why Mantu test is not done currently to actually check for TB, but it was a very preliminary type of investigation which was done earlier, right? And of course, you are right that this can be used to determine whether the immune cells are working or whether the immune system is working properly. Yes. So, any queries till this? If not, then we will move on to the question answers. Please tell me your answers. Any other answer? Yes. So the correct answer is B. It is an example of type 2 hypersensitivity. Okay. Next question. Right. So the correct answer is B. It is the IgE mediated type 1 hypersensitivity reaction where the release of vasoactive amines are observed. Okay, next question. Yes. Yes, the correct answer is D, all of the above. All of the above events are associated with the release of histamines. Um, can you tell me what will be the order of these events? Can anyone tell me? Like, among these, which one comes first, which one comes second?
ABC, cross-linking of Ig with the allergen, phosphorylation of uh, phospholipase C by protein tyrosine kinase. Protein kinase C and calcium ions help in microtubule contraction.
Exactly.
ABC,
So
basic deficiency.
exactly, just in the order it is mentioned, right? Okay. So this is the correct answer, all of the above. Okay. Moving on to the next question. Okay. The correct answer is the T cell mediated hypersensitivity is type 4. Right. Okay, and the last question for today. Yes, the correct answer is ROGAM. Alright, so just to summarize these hypersensitivity reactions once again, the four types of hypersensitivities. Type 1 is mediated by IgE, okay, and IgE helps in release of the vasoactive amines from the mast cells or the basophils, which causes this inflammation, right? Type 1 hypersensitivity is the most common type of hypersensitivity which can be prevalent in the cold allergy, dust allergy, or the like pollen allergy. And type 2 hypersensitivity reaction is mediated by the complement pathway or the antibody dependent cell mediated cytotoxicity. So either there is a killing of the cells or the activation of the complement pathway. And one example, classic example, is the erythroblastosis fetalis. Next is the type 3 hypersensitivity. Type 3 hypersensitivity is mediated by the antigen antibody complexes. It is initiated by the antigen antibody complexes, and it is mediated by the activation of the complement pathway. And the complement pathway actually activates the inflammation. It enhances the inflammation, and also it causes the more and more extravasation of the neutrophils to the site of action, which causes a local or a global type of response in type 3 hypersensitivity. Whereas in type 4 hypersensitivity, it is different from the rest because it is mediated by the T cells, and T helper cell 1, Th1 cell is initiates this type 4 hypersensitivity, where the activated macrophages, they produce several cytokines which initiates this which elicits this kind of hypersensitivity reaction. And type 4 hypersensitivity is also a delayed type hypersensitivity because it takes time to activate the macrophages, which elicits the hypersensitivity reaction, right? So that's all for today. Do you have any other questions?
Ma'am?
Yes, one by one.
Okay, ma'am. Cytokine stop is completed, ma'am.
Sorry, I did not get you.
Cytokines, ma'am.
um,
Yes, it was covered earlier. It is done now.
Madam, in the
yes.
previous week, that is 10th week, first question, you
Ninth
told
week.
that all the complement pathways, the serine protein proteases, all C3 convertases. Madam,
No, the serine proteases are involved in cleavage of the complement proteins. Several complement
yes, madam.
proteins. Yes.
You told that all C3 convertases are not serine proteases earlier.
No, not the C3 convertases. The in the lectin binding pathway, there is these serine proteases which are involved, which are membrane associated serine proteases. But all the not necessarily all the cleavage that happens occurs by serine proteases, but there are several serine proteases which are involved in several complement pathways, like the alternative pathway, the lectin pathway, as well as the classical pathway.
Okay, ma'am. Okay.
Okay, anything else for today?
Compliment for you, ma'am. Very well explained. A step ahead. I think ROGAM was not covered in the class also.
No,
Thank you so much.
it was covered in the class. You might have missed it.
Okay,
Covered,
then I
madam.
must have missed
That's
it.
covered.
Thank you so much.
Yes, it was covered. Okay. So thank you all for joining today.
Ma'am.
Yes, Rajkumar, go on.
Ma'am, last class is cytokines introduction only, ma'am. Full classes completed, ma'am. Cytokines.
The cytokines. Yes, this
Yeah.
cytokines, there was this introduction about cytokines, but in this class, I actually focused on the hypersensitivity part more, and so I did not take in consideration the cytokines other lectures. If you want any help, you can consider the other there is another TA with me, Shimanti. She actually, I think, discussed the cytokines part in her week 10 class. So if you want to go through that, you can, or if you want any other help specifically, then you might post in the group. I will see to it in the next session.
Okay, ma'am. You told ma'am cytokines your topic in
Okay, okay.
last class. Okay, thank you, ma'am.
Thank you. All right. Thank you so much for joining today. We will meet again on the next class next Tuesday.
Ma'am, one more thing,
Yes, 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 yes.
ma'am.
Go on.
Ma'am, off the record, out of the 12 weeks, if I want to study only 8 weeks, which course should I leave?
Okay,
Because
let
this
me
is
let me finish
Diwali time, ma'am. It is
let me
not
get over with the
possible.
recording
I'm
first.
asking